Development of the teeth. We will now discuss the histologic aspect of tooth development and the coming together of different tissues that form the tooth and its surrounding. For every developmental event, a complex and intricate cascade of gene expression takes place. This is to direct cells to the right place and onto the proper differentiation pathway. After about 37 days, a continuous band of thickened epithelium, referred to as odontogenic epithelium, forms around the mouth in the upper and the lower jaws. They are horseshoe-shaped, and they correspond in position to the future dental arches of the upper and lower jaw. The formation of these thickened epithelial bands is the result of increased proliferative activity within the epithelium, as well as the change in orientation of the mitotic spindle and the cleavage plane of the dividing cells. Each band of epithelium, called the primary epithelial band, quickly gives rise to two subdivisions. Firstly, the dental lamina, which forms first, and then the vestibular lamina, which forms shortly afterward and which is positioned just in front of the dental lamina. A key feature of the initiation of tooth development is the formation of localized thickenings within the primary epithelial bands. On the anterior aspect of the dental lamina, continued and localized proliferative activity leads to the formation of a series of epithelial outgrowths into the ectomesenchyme. These are at sites that correspond to the positions of the future deciduous teeth. The ectomesenchymal cells accumulate and they then condense around these epithelial outgrowths. From this point, tooth development proceeds in three stages: the bud, the cap, and the bell stages. These terms describe the morphology of the developing tooth germ, but do not describe the functional changes that occur during development. The vestibule forms as a result of the proliferation of the vestibular lamina into the ectomesenchyme soon after the formation of the dental lamina. The cells of the vestibular lamina rapidly enlarge and then degenerate to form a cleft that becomes the vestibule between the cheek and the tooth-bearing areas. Determination of tooth type. The determination of specific tooth types at their correct positions in the jaws is referred to as patterning of the dentition. Two hypothetical models have been proposed to explain how different tooth shapes are determined, and evidence exists to support both. The bud stage is represented by the first epithelial ingrowth into the ectomesenchyme of the jaw. The epithelial cells show little change in shape or function. The supporting ectomesenchymal cells are packed closely beneath and around the epithelial bud. As the epithelial bud continues to proliferate into the ectomesenchyme, cellular density increases immediately adjacent to the epithelial outgrowth. This process is referred to as condensation of the ectomesenchyme and results from a local grouping of cells that have failed to produce extracellular substance and have thus not separated from each other. As the tooth bud grows larger, it drags along with it part of the dental lamina. The epithelial outgrowth, which superficially resembles a cap sitting on a ball of condensed ectomesenchyme, is referred to as the dental or enamel organ. This will eventually form the enamel of tooth. The ball of condensed ectomesenchymal cells, called the dental papilla, will form the dentine and the pulp. The condensed ectomesenchyme encapsulating the enamel organ is called the dental follicle. This gives rise to the supporting tissues of the tooth. Because the enamel organ sits on the top of the dental papilla like a cap, this stage of tooth development is known as the cap stage. The enamel organ, dental papilla, and dental follicle together constitute the tooth germ. Important developmental changes in terms of histodifferentiation begin late in the cap stage and continue through to the bell stage. The epithelial cells transform themselves into morphologically and functionally distinct components. The cells in the center of the enamel organ synthesize and secrete glycosaminoglycans into the extracellular compartment between the epithelial cells. These glycosaminoglycans are hydrophilic, and so they pull water into the enamel organ. The center of the enamel organ is termed the stellate reticulum because the cells assume a star shape due to them being forced apart by the glycosaminoglycans and the water. Continued growth of the tooth germ leads to the next stage of tooth development, the bell stage. During this stage, the tooth germ assumes its final shape, a process referred to as morphodifferentiation. This process determines the final shape of the tooth crown. The cells that will make the hard tissues of the crown, that is the ameloblasts and the odontoblasts, acquire their distinctive phenotype. This process is referred to as histodifferentiation. At the periphery of the enamel organ, the cells assume a low cuboidal shape and form the outer enamel epithelium. The cells bordering the dental papilla assume a short columnar shape and are characterized by high glycogen content. They form the inner enamel epithelium. The outer and the inner enamel epithelia are continuous, and the region where they meet is termed the cervical loop. This is at the rim of the enamel organ. In the bell stage, some epithelial cells between the inner enamel epithelium and the stellate reticulum differentiate into a layer called the stratum intermedium. The cells of this layer are soon characterized by an exceptionally high alkaline phosphatase enzyme activity. Although these cells are histologically distinct from the inner enamel epithelium, both layers work synergistically and have been considered as a single functional unit responsible for the formation of enamel. The enamel organ is supported by a basal lamina around its periphery. The dental papilla is separated from the enamel organ by a basal lamina from which a mass of fine fibrils extend into an acellular zone, and it is here that the first secreted enamel matrix proteins accumulate. The dental papilla is referred to as tooth pulp when the first calcified matrix appears at the cuspal tip of the Bell stage tooth germ. Two other important events occur during the Bell stage. The dental lamina fragments eventually, separating the tooth from the oral epithelium. The inner enamel epithelium completes its folding, hence making it possible to recognize the shape of the future crown pattern of the tooth. The folding that occurs as the crown develops results from intrinsic growth caused by differential rates of mitotic division within the inner enamel epithelium. The cessation of mitotic division within the cells of the enamel epithelium determines the shape of a tooth. When the tooth germ is growing rapidly during the cap to the Bell stage, cell division occurs throughout the inner enamel epithelium. As development continues, cell division ceases at a particular point because the cells are now beginning to differentiate and assume their eventual function of producing enamel. The point at which the inner enamel epithelial cell differentiation first occurs represents the site of the future cusp development. This is because the inner enamel epithelium is constrained between the cervical loop and the cusp tip. Continued cell proliferation causes the inner enamel epithelium to buckle and to form a cuspal outline. Thus, the future cusp is pushed up towards the outer enamel epithelium. Eventually, differentiation of the inner enamel epithelium and papilla cells sweeps down along the cusp slopes and is followed by the deposition of dentine and enamel. These two matrices are deposited face to face, thereby defining the dentino-enamel junction, or the DEJ. The occurrence of a second zone of cell differentiation within the enamel epithelium leads to the formation of a second cusp. A third zone leads to a third cusp, and so on until the final cuspal pattern of the tooth is determined. Pioneer nerve fibers approach the developing tooth during the bud to cap stage of development. The target of these nerve fibers is the dental follicle. Nerve fibers ramify and form a rich plexus around the tooth germ. The nerve fibers only penetrate the dental papilla once dentinogenesis begins. Although a possible relationship has been assumed between the developing nerve and blood supply, the timing differs in establishment of the papillary vascular and neural supplies. The initial innervation of the developing teeth is concerned with the secondary innervation of the future periodontal ligament and pulp. At no time do nerve fibers enter the enamel organ. Fragmentation of the dental lamina results in the formation of discrete clusters of epithelial cells that normally degenerate. Some may persist and are given the name epithelial pearls. These clusters of cells may form cysts referred to as eruption cysts over the developing tooth and delay eruption. They may also give rise to odontomes or may be activated to form supernumerary teeth. An important consequence of the fragmentation of the dental lamina is that the tooth continues its development within the tissues of the jaw, separate from the oral epithelium. But before the tooth can function, it must re-establish a connection with the oral epithelium and penetrate it in order to reach the occlusal plane. This penetration of the lining epithelium by the tooth is a unique example of a natural break in the epithelium of the body. Integrity is re-established by the formation of a special seal around the tooth referred to as the junctional epithelium. So far, only the initial development of the deciduous dentition has been described. The permanent dentition also arises from the dental lamina. The next step in the development of the tooth is terminal differentiation of ameloblasts and odontoblasts and the formation of the two principal hard tissues of the tooth. This process is referred to as histodifferentiation. Until the crown assumes its final shape during the cap and early bell stage, all cells of the inner enamel epithelium continuously divide. Thereafter, until the tooth crown attains its full size, only cells of the cervical margin of the enamel organ divide. As morphologic changes occur in the cells of the inner enamel epithelium, changes also occur within the adjacent dental papilla. The undifferentiated ectomesenchymal cells increase rapidly in size and ultimately differentiate into odontoblasts, which are the dentine-forming cells. These cells differentiate into odontoblasts under the organizing influence from the cells of the inner enamel epithelium. Note that in the absence of epithelial cells, no dentine develops. The ectomesenchymal cells of the dental papilla assume competence only after a set number of cell divisions. As development continues, progressive differentiation of the cells of the inner enamel epithelium down the cusp slopes and differentiation of odontoblasts in the papilla takes place. The odontoblasts, as they differentiate, secrete the organic matrix of dentine, which then ultimately mineralizes. As the organic matrix is deposited, the odontoblasts move towards the center of the dental papilla, leaving behind a cytoplasmic extension around which dentine is formed. Just before the first layer of dentine forms, referred to as mantle dentine, differentiating inner enamel epithelial cells become ameloblasts, and they secrete some enamel proteins. These first proteins, together with growth factors, play a role in the epithelial mesenchymal signaling that leads to the terminal differentiation of the odontoblasts. This is due to possible interaction with components of the basal lamina that separates them. Before forming the first dentine, cells of the enamel organ, and in particular those of the inner enamel epithelium, receive nourishment from two sources: blood vessels located in the dental papilla and vessels situated along the periphery of the outer enamel epithelium. When the dentine forms, it cuts off the papillary source of nutrients, causing a drastic reduction in the amount of nutrients reaching the enamel organ. This reduction occurs when the cells of the inner enamel epithelium are about to secrete enamel, and thus the demand for nutrients increases. This demand for nutrients is satisfied by an apparent collapse of the stellate reticulum and invagination of the outer enamel epithelium by blood vessels lying on the outside. Root formation. The root of a tooth consists of dentine covered by cementum. It follows from the previous discussion that epithelial cells also may be required to initiate the odontoblasts, which eventually form the dentine of the root. Epithelial cells of the inner enamel epithelium and outer enamel epithelium proliferate from the cervical loop of the enamel organ to form a double layer of cells known as the root sheath of Hertwig. This sheath of epithelial cells extends around the dental pulp, between the pulp and the dental follicle, until it encloses all but the basal portion of the pulp. The rim of this root sheath, the epithelial diaphragm, encloses the primary apical foramen. As the inner enamel epithelial cells of the root sheath progressively enclose more and more of the expanding dental pulp, they initiate the differentiation of odontoblasts from ectomesenchymal cells of the periphery of the pulp that faces the root sheath. These cells will eventually form the dentine of the root. A single rooted tooth is formed in this way. In the case of mandibular teeth, the tip of the forming root remains in a basically stationary position relative to the inferior border of the mandible. This means that the free border of the root sheath must be in a stable position with the onset of root formation. The crown of the tooth is growing away from the bony base of the crypt, and the root sheath is not actually growing into the jaw. Because of these growth changes, the root sheath is stretched. Although cell division occurs within it, it eventually forms a number of discrete clusters of epithelial cells separated from the surrounding connective tissue by a basal lamina. These are referred to as the epithelial cell rests of Malassez. In adults, these epithelial cell rests persist next to the root surface within the periodontal ligament and are apparently functionless. The developmental biology of the tooth root is the least studied aspect of tooth development in terms of gene expression and signaling molecules.